Hello, everyone, and welcome to the next installment in our series, Dr. Gilbert Hosts. My name is Dr. Rebecca Gilbert, and I am the Chief Scientific Officer at American Parkinson Disease Association. Thank you so much for joining us today. I am particularly excited about today's guest who is with us, Dr. Walter Koroshetz. Dr. Koroshetz is the director of one of the branches of the National Institutes of Health, or the NIH. The branch that Dr. Koroshetz directs is the National Institute of Neurologic Disorders and Stroke, or the NINDS, which focuses on neurologic disorders, including Parkinson's disease. In this capacity, Dr. Koroshetz oversees more than $150 million in Parkinson's disease research funding. Therefore, Dr. Koroshetz is one of the most influential people in the country and around the world on how Parkinson's disease research is conducted and funded. An APDA, as part of the Parkinson's disease community, has access to him today to hear firsthand about Parkinson's disease research at the NIH and to ask him our questions. Dr. Koroshetz will start with a short presentation about PD research at the NIH, and then we'll open up this session to your questions about the NIH and the NINDS. American Parkinson Disease Association, or APDA for short, is the largest grassroots network dedicated to fighting Parkinson's disease and works tirelessly to assist the more than 1 million people with Parkinson's disease in the United States live life to the fullest. Please visit our website at apdaparkinson.org to explore all that we have to offer. And you can also contact APDA through our toll-free helpline, 1-800-223-2732, as well as via our social media outlets. And now I'd like to formally introduce my guest. Before joining NINDS, Dr. Koroshetz served as Vice Chair of the Neurology Service and Director of Stroke and Neurointensive Care at Massachusetts General Hospital. He was Professor of Neurology at Harvard Medical School. Dr. Koroshetz joined NINDS in 2007 as the Institute's Deputy Director and became Director in 2015. Dr. Koroshetz has held leadership roles in a number of NIH and NINDS programs, including the NIH's Brain Initiative, NIH Blueprint for Neuroscience, and the Helping to End Addiction Long-Term or HEAL initiative. I am so excited to be talking with Dr. Koroshetz today. Dr. Koroshetz, welcome, and thank you so much for joining us.
Thank you very much, Rebecca. It's a real pleasure. And the APDA has been a great partner with NINDS and the work that we're trying to do to reduce the burden of illness due to Parkinson's disease. And I just want to start off by acknowledging what must have been, a very tough year for folks with Parkinson's and their caregivers, given the COVID epidemic and the effects it's had on, getting the care you need. But hopefully things are improving and you can again get the kind of care that you've been used to. So I'm going to talk today about kind of where we are in terms of Parkinson's disease research, and the NIH is the taxpayers' investment in biomedical research, and at NINDS we're responsible for a couple of hundred, some people say 400 different neurologic disorders, and Parkinson's disease research has been a really important part of our portfolio for many decades. I want to talk about the major goals of Parkinson's research and kind of set the stage here. So right from the bottom, let's go from the bottom. What we want to do is, of course, reduce the symptoms that people have that cause them disability or annoyance or stop them from doing the things they want to do. And that's what we call symptomatic therapies. And the good news in Parkinson's disease is that symptomatic therapies are really quite effective in the early stages of the illness. So dopamine replacement with Sinemet, levodopa, carbidopa, or other dopamine agonists can really take away symptoms in the early stages, particularly the motor symptoms in the early stages of disease. And then we have deep brain stimulation, which is also very effective as the medicines start to wane in their effectiveness. and potentially even, for some people who have a hard time tolerating the medicines, can be useful even in the earliest stages. And deep brain stimulation is actually really quite interesting. It's manipulating the circuits in the brain that are responsible for the motor signs of Parkinson's disease. And luckily enough, scientists over the years, working initially with non-human primates and then moving into people, were able to show the effectiveness of deep brain stimulation. It turns out now that the most interesting aspect of this is going on in concert with what we call the Brain Initiative at NIH. That stands for Brain Research for Advancing Innovative Neurotechnologies. And it's all about trying to learn about how circuits work. And so the circuits involved in Parkinson's are a target for many of the studies, but it's particularly for the studies that involve humans. So patients with Parkinson's have been very valuable in volunteering for new studies to learn how to monitor and then modulate brain circuits. And a new advance that's coming out shortly is an actual new type of stimulation where the degree of stimulation is actually controlled by the patient's own brainwaves. So as opposed to what we have done for years is to set the stimulator at a certain degree of settings and then see how things go. In the new world, those settings might be changing minute to minute, day to day, depending on the needs of the patient, as illustrated by actual brainwave activity that can be captured with these devices. So it's called a closed loop brain stimulation to reduce the symptoms, particularly the motor symptoms of Parkinson's disease. Now, in contrast to our ability to look at symptomatic therapies, there has been a big push over the last 10 to 20 years to try and develop therapies that would stop the disease or slow down its progression. And this has not really reached fruition now, but it's kind of one of the major goals of much of the research that we fund. And there are some clues that people are following, which do seem very promising. I want to say right from the beginning that what we now know about Parkinson's disease is that it is a form of a synucleinopathy. Synuclein is a protein. And it turns out that in Parkinson's disease, the signature are aggregates of synuclein in what are called Lewy bodies. The Lewy body is kind of like a ball. And what was discovered was it's filled with synuclein. Synuclein is a protein. And for some reason in Parkinson's disease, the nerve cells have trouble metabolizing the synuclein, and instead it forms aggregates, tangles that then form into these balls, the Lewy bodies. And that is considered to be the main culprit for Parkinson's disease. And so the big questions are, how does this start? How does this synuclein aggregation begin? What triggers it? And how to stop it? What we do know now from the last, say, about 5 years worth of work is that synuclein aggregates spread from cell to cell. So the most exciting stuff now is understanding how synuclein aggregation in one cell then leads to synuclein aggregation in a neighboring connected cell, and that one to another connected cell, and how it spreads throughout the nervous system. And we'll talk a little bit about that down the road. But the key point is that if we can figure out how to prevent the spread, then we should be able to prevent the progression of the disease. And this has been influenced heavily by the genetic studies that identify mutations in genes that cause Parkinson's. We'll talk about some of these down the road, but the first one was actually a mutation in the synuclein gene itself. So in almost any mutation where you get an increase of synuclein production, you get Parkinson's disease. Now, if we are successful at developing a therapy that slows down the progression of disease, then that leads us to the potential to actually prevent the disease. And the reason for that is that what we've learned in the last 10 years or so is that this synuclein spread probably starts outside the nervous system, outside the central nervous system, outside the brain. One of the greatest potential sources of the beginnings of Parkinson's is the aggregation of synuclein in the nerve fibers that go to the gut. So, for instance, there are studies now that have identified certain bacteria that will cause synuclein to form aggregates. And these bacteria are in your gut, and there are nerves that are in contact with the gut. So it's possible, this is a hypothesis, that the synuclein could start there and then move through those nerves, which then go into the spinal cord and the brainstem, and then they could spread from that site up into the brain to give the symptoms of Parkinson's, and the symptoms keep getting worse as the synuclein spreads throughout the brain. Now, if we can develop a disease-modifying therapy, we could potentially find this synuclein aggregation occurring in the gut, say, or even in the spinal cord, and stop it before it causes any symptoms whatsoever. So that would be preventing the disease. And it all follows from identifying a disease-modifying therapy. And then one would ask, well, how could you know who has synuclein in the gut or has these early stages? Well, it turns out that many people with Parkinson's will have some symptoms, loss of sense of smell or constipation or trouble with sleeping. And these occur 10 years before the symptoms occur of Parkinson's, the motor symptoms. So therefore, there's a lot of effort trying to identify what are the signatures that indicate someone will develop Parkinson's in the future. And those are actually looking quite promising. That if we had a disease-modifying therapy, we could actually test it in people who have these early symptoms, which are not that disabling at all, but could, if we treat them with effective therapy, may even prevent the known symptoms, the motor symptoms of Parkinson's from ever occurring. So that's kind of it in a nutshell of what we're trying to do. Now, it's actually all complicated, and it's all, a lot of speculation, a lot of gaps in knowledge that have to be filled in for us to be successful. But we do have resources at NIH, and let's talk about the NIH investment in Parkinson's disease research. So as you can see here on the slide, the actual amount of funding going into Parkinson's research has increased pretty much steadily since 2016. The budget at NIH was pretty flat before that, but we've had increases since. And then you can see that NINDS is not the only institute that's studying Parkinson's. There are other institutes like the Aging Institute, which has a large amount of funding for neurodegenerative diseases, and the Environmental Health Institute, NIEHS, because as we mentioned, there's a lot of interest now in how the synuclein aggregation occurs, what triggers it, and the triggers are thought to be something related to our environment, say, like I mentioned, bacteria, but there could be other things, toxins, things that we're exposed to that get this process started. So there are other institutes involved. And now a large amount of the increased funding has come to NIH because Congress appropriated funds to the Aging Institute to try to reduce the burden of dementia in the US. And NINDS manages about 10% of those funds for particular areas of dementia, one of which was identified by Congress as Lewy body dementia. Now, I mentioned Lewy bodies. Those are those globs of synuclein that are the signature of Parkinson's disease, but they're also the signature of what's called Lewy body disease. And one could think of Lewy body disease as identical to Parkinson's, except it starts out its symptoms by affecting the brain thinking circuits as opposed to the motor system. And the motor system comes second. In Parkinson's disease, as we know it, the motor symptoms come first, and then the thinking problems and cognitive problems come later in the disease. But the good news is with this increased funding, we've been able to use a considerable amount of it to study the processes that are common to both issues. For instance, like how synuclein spreads or how the synuclein causes trouble. That's pertinent to both Parkinson's and Lewy body disease. So the NIH approach to synucleinopathy dementia research on the next slide indicates the kind of research that's been able to be done in this space and is responsible for a considerable proportion of the increase in funding for Parkinson's disease because of this overlap of Alzheimer's disease and Lewy body disease funding with Parkinson's disease funding, as is seen on the next slide, which shows the growth from 2015 to 2019. AD is Alzheimer's disease, AAD-related dementias includes Lewy body disease. So you can see here the overlap that considerable amount of funding have come from this appropriation from Congress to help us better understand Parkinson's disease as well. And and this has really helped. So if we see on the amount of funding going to Lewy body dementia, which also includes the cognitive troubles that come in Parkinson's disease, you can see the growth that has occurred over time due to this funding that's available through the appropriation to Congress for dementia research. And there are lots of programs that have come from this. So we're doing using really cutting edge tools to look at post-mortem tissue from patients with Parkinson's disease. There are new technologies now where we can look at every single cell in the brain and try and see how it's been affected by Parkinson's disease, and that can give clues as to targets for therapies going forward. And just going to some of the studies which we think are really quite interesting. We talked about synuclein aggregation and how it spreads. And this is an example of a study from Johns Hopkins, from Ted Dawson's lab, where in animals, they're able to inject synuclein that comes from patients with synucleinopathy, and you inject it into the intestinal wall of the animal, and then they were able to see it spread over time into the brain, into the hippocampus or the prefrontal cortex or the amygdala or even the nigra. And so it's almost, this spreading model is now available for testing. And what they did was they were able to cut the nerves from the gut to the spinal cord, and that prevents the spread into the brain. Of interest, there are reports that patients who underwent vagotomy for ulcer disease, not uncommon practice 30 years ago, that they actually had less chance of getting Parkinson's disease. Not a lot of numbers there, but there is a suggestion that this actual experiment that they did in the animal, the mouse here, has actually is a replication of something that happened in humans 30 years ago. Now, synuclein, we would really like to be able to see it so that we know where it is and that we can develop treatments to make it go away, these aggregates. And so techniques using high energy cryo-electron microscopy are able to look at the actual aggregates themselves at these detailed molecular level using electron microscopy. this is an example looking at the protein tau. It's not synuclein, but it's the same idea that we have this study, which we fund to people to do these cryo-electron structures of tau and synuclein to understand how they're put together. And this would allow new drugs to dock into these aggregates and potentially help to have them removed from the body. It also would help us to understand how proteins could dock so we could radio label them and then have a scan, a PET scan, that would tell us where the synuclein is. And this would be incredibly valuable in developing therapies to remove synuclein from the brain. And so these are the kind of things that people are doing now, things that could never be done, even 5 years ago. We also have a very interesting partnership with industry and other agencies and nonprofits, particularly the Michael J. Fox Foundation. And in this project, we all come together to bring the data from patients, people who are listening on this talk, who have participated in, say, PPMI project or the BioFind project at NIH, or some of the NIH clinical trials, and the Harvard Biomarker Consortium studies. And all these data from all the patients, the clinical data is all brought together along with whole genome sequencing, protein studies from the blood, gene studies from the blood, metabolic studies from the blood. Just an incredible treasure trove of data brought together, all linked together, available for anyone from around the world to come in and examine it for clues as to, say, the progression of Parkinson's or biomarkers of Parkinson's compared to controls. So the AMP-PD knowledge platform components are the clinical, the genome, the RNA transcriptome, and this is all on an AMP-PD knowledge platform architecture. And our plan now is to put this out. It is out, actually, ready, but to incentivize people to come in and use their machine learning or artificial intelligence tools to examine this data in detail using the platform architecture as seen on the next slide. And that platform architecture is put together by the Verily Corporation, which is an offshoot of Google. So it's quite a sophisticated database for people to look into. So things are moving very quickly, lots of excitement. We're hopeful that some of the trials that are now ongoing may come to fruition and give us clues, but we really have to take the long road approach and persist. I think persistence is really the most important thing. And we certainly appreciate all the effort that all the patients with Parkinson's have put in to enable this research to be done, because the real clues come from the patients. So thank you very much. I'm happy to answer any questions.
Thank you so much, Dr. Koroshetz, for that excellent introduction. And as Dr. Koroshetz just said, we are now ready for our Q&A. Now, we have a number of questions, actually a lot of questions that were submitted to us during registration, but we also encourage you to submit your questions live right now, whether you're on Facebook or YouTube. In order to participate in the live chat, you'll have to log into YouTube or Facebook, and we may even display your question along with the name and photo you're logged in with. So please keep your questions focused on the NIH so we can best use Dr. Koroshetz's expertise. So let's start with the first question. This is a question from Roger. When will the NIH increase the current funding of $200 million a year for Parkinson's disease? And he compares it to the $3 billion that the NIH spends on HIV, for example.
Okay, well, good question, Roger. So the $3 billion for HIV was actually mandated by Congress for many years. That's how that number got there. We don't have a mandate for spending on Parkinson's disease. Instead, the amount of funding that goes to Parkinson's disease, as is true about all our diseases, 200, 300 diseases, depends on what grants come in to NINDS. And so the more grants that come in that get scored highly meritorious, the more money goes to Parkinson's. And the less grants come in, the less money goes to Parkinson's. It goes to another disease area or another neuroscience area. So that's kind of how the budget works. Now, the difference today is that there is congressionally mandated funds that I talked to you about for dementia research. And that is about $3 billion a year now for dementia research. And as I mentioned, because synucleinopathy causes Lewy body disease and Parkinson's disease, and biologically they're the same disease, we're able to use a considerable amount of that funding as well for the Parkinson's research. So that has really given us an increase, and that's mandated by Congress. So that allowed us to put out a number of different projects that are looking at synucleinopathies, and that's additional funding. So I hope that explains how the budgets work. But at NIH, it's a very bottom-up process for the budgets, unless there's some kind of congressionally targeted project that Congress tells us to do and gives us money for.
So along those lines, Ian, who is a medical researcher, had a question. So you mentioned that Congress has mandated a certain amount of money for dementia research, and that would include Lewy body dementia or dementia that is associated with Parkinson's disease. But since dementia is prevalent in Parkinson's disease, why doesn't that money not encompass all of Parkinson's disease basic research, given that advances made in basic research about Lewy bodies in general or about Parkinson's disease in general could also impact dementia?
Right.
What do you think about that?
Yeah, he's right. He's actually right. That's what happened.
Okay.
So any of the basic science work that one would do on Parkinson's disease. So if you want to know how synuclein spreads, what does synuclein do to the nerve cells, that is all relevant to Lewy body disease. The difference would be, it's kind of technical, is that if your grant was focused on only the motor neuron or the nigral cells, that might put you out of what would be considered dementia research. So, but people have learned, these investigators are very smart people. What they've learned is to always say that, this is the research, it's relevant to Parkinson's disease, it's relevant to the dementia in Parkinson's, it's relevant to Lewy body disease. And then there's basically a computer that codes the grants based on what they say, and that classifies them into what's called Alzheimer-related dementia or not. And that's basically how it works. So I think the only downside is that, it's a lot harder to qualify that kind of research if you're really looking just at the motor sides of Parkinson's. But pretty much everything, all the other aspects and all the basic science that underlies those aspects are fair game for this funding.
Okay, that's great information for our researchers.
Yes.
So you want to make sure to highlight in your grant to the NIH that this has relevance for alpha-synuclein in general and potentially for dementia in general, and that gets you potential access to this congressionally mandated money. So that's really good.
That's right.
That's great information.
Right. So I'd say that just to add that, so at NIH, as I was just talking about, the grants come in, they get reviewed, they get scored, and then we fund the best scored grants until we run out of money. Pretty simplistic process, but for our grants now, we run out of money when we fund the top 14%. So that means that 86% don't get funded. But if you qualify as doing synucleinopathy or Lewy body disease work or dementia in Parkinson's or cognition in Parkinson's, you qualify as ADRD, then that's 28th percentile chance of getting funded. So you double your chances if you write a grant that fits in this space.
Okay, fantastic. We have a lot of questions about clinical trials. Michael asked a question: how do people with PD find out about clinical trials, specifically being conducted at the NIH about Parkinson's disease?
Okay. So there is all trials done in the U.S. have to be registered under clinicaltrials.gov. So any trial going on in the U.S., whether it's industry or NINDS funded, it's going to be there. And NINDS has a page which links people to the clinicaltrials.gov for Parkinson's trials. And then I know that some of the organizations, I know Michael J. Fox has a trial finder app where you can find out what trials are going on in Parkinson's and who's sponsoring them, whether it's industry or NIH. And we also have, should have mentioned, maybe that's what the question is, we also have in NIH funding, 90% of the money goes out to universities around the country. About 10% of the funding stays to run the operation of NIH, and a good proportion of that goes to fund research on a campus in Bethesda. And we also have a very strong group in Bethesda studying Parkinson's disease and Lewy body disease. And so they're also doing research in the Bethesda area. They have a website for the Parkinson's research that they're doing there. So that's another option.
Okay, fantastic. Thank you. We have a question from Paul. It's a two-part question. So he has participated in multiple research projects over time. And what are your thoughts about requiring researchers to share the results with participants or with general public? What are your thoughts on that?
Right. So NIH has a data sharing policy that strongly encourages data sharing for all the research that we fund. For all our clinical trials, we bring the data in, make it available to people. And the issue that was brought up is, I think, a really important one, and that is letting the patients know about the trial and the trial results. And so that's something that I'm actually working on now at NINDS, to do that more, and not only share at the end, but actually try and have patients involved in the trial as they're beginning, as they're designed, as they're executing. Because I think looking around at what I've seen from the other institutes, there's some really good examples where including the patients and these patient engagement, basically having them as part of the team of clinical trials has had tremendous influence on helping the trials get started and completing, making sure that the investigators don't make it so onerous that nobody in the right mind could ever do this trial. The patients will be able to tell the investigator that before the investigator figures it out. But that's just one example. Communicating to patients about a trial. Sometimes doctors are just not very good at that. But a patient who's been working with the trial can really, get on the same level as that patient who's thinking about joining the trial and know what their concerns are, because that person's been through it before. So I think that's something we're really trying to do more. Now, I would say that NIH legally is bound by what's called the Bayh-Dole law. And that law stipulates that NIH funding research findings belong to the people who got funded, the institution who got funded. And so if an institution does research and they find something and they want intellectual property, they can't make it public. Right now, with that law, NIH can't really do anything about that. But that may also be important, because then universities can develop the IP, work with companies, start a trial. So there's two sides to that. You can't develop a therapy without intellectual property. So we're always caught between the sharing and the danger of damaging the intellectual property for a treatment.
Thank you for that. And I just want to mention that anytime I've interacted with the NIH and NINDS, there's always been patient involvement. So that's really been a theme. And I think that's a wonderful thing about the NINDS is that there's a real concern to include patients in every step of the way and in presenting results and in developing the sort of vision. So want to thank you. Thank you for that, for including the patient voice and for being here today to speak to patients themselves.
Well, I mean, one example is, the patients, this was probably maybe 5 or 6 years ago, we were hearing from the patient groups that that we were ignoring the non-motor signs of Parkinson's disease. We were concentrating on the motor signs. So that actually led us to a shift to understand the non-motor sign. And what do you know? Now that allows us to use all this funding that came for dementia research from Congress to look at the non-motor signs. So yeah, it's good to pay attention to patients.
Absolutely. Absolutely. So that actually leads to a question that a couple of researchers have asked, which is, so you mentioned that NINDS has a certain budget. If it's not the ADRD mandated money, then it's the sort of general pot of money. And you basically assess which grants come in and you give it out to the meritorious grants. And there is no sort of disease-specific selection in that process. So if more people give grants for Parkinson's disease or other diseases, then it will be distributed based on what grants you see. So are there any opportunities for specific RFAs within Parkinson's? So one researcher wanted to know, is NIH or NINDS interested in research into astrocytes in Parkinson's? Another wanted to have more money on non-motor, as you mentioned. So is there any leeway within the NIH or NINDS, I should say NINDS specifically, to give money to specific ideas within Parkinson's or within a specific disorder, or is it really just whatever comes in is what you have to deal with?
So good question. So we do, we're always listening to the scientists and trying to identify gaps. And when we identify a gap, then we will consider putting out an RFA to fill a gap. That's kind of the strategy. And so, so for instance, well, the issue of the astrocytes and the changing from a loving astrocyte in the brain to an astrocyte that's activated, it could be causing damage. Turns out that that's a big problem that for many different diseases. And so, in fact, for that, all the neuroscience institutes got together and we actually put together an RFA just to look at that problem in the brain as a generic problem. Which gets me to the other point, which I'd say is that the answers that are going to be helpful to Parkinson's disease, they're coming, but I don't know where they're coming from. And I would bet that the real thing that causes the breakthrough is some discovery that comes from outside of what's Parkinson's disease research. So if you think about the neurodegenerative diseases, they share so many things in common, as was just mentioned. What's the role of the astrocytes? What's the role of inflammation in the brain? This is the biggest question going on right now in neuroscience for many diseases. So the answer might come from Alzheimer's that helps Parkinson's, or from Parkinson's that helps Alzheimer's. In terms of the gaps, so the big gap that we went after, we started many years ago, is biomarkers in Parkinson's, because for a drug company to invest in a neurodegenerative disease, they need to have something they can measure before they go into these ultra expensive phase 3 trials. So the best example is synuclein. So you want a drug that's going to reduce synuclein. That's kind of what's right front and center. But if you have a drug and you have 100 patients and you have nothing to measure, if you have to wait and look at the progression of disease, which can take 2 or 3 years to figure out, they can't go. It's a no-go for the company. If they can measure synuclein and then use a drug decrease the amount of synuclein, say in the spinal fluid, and that's big business. That gets them into the area. So we invested heavily in the biomarker. So we have, and as did Michael J. Fox, and that's what a lot of the AMPD data is coming from. And then we also continue to fund biomarker work in Parkinson's disease. And I would say that there's something that really interesting has come out of it, and that's a test that looks incredibly accurate at detecting synuclein. It can be detected from the spinal fluid. And it actually came from work that was done on another disease called Creutzfeldt-Jakob disease, which is a prion disease. And prion disease the basis is a protein that aggregates and spreads throughout the brain. And so people out at the NIH in Montana are working on tests, to be able to screen cows. It's the same thing, mad cow disease, it's a prion disease. So they came up with an assay where if you take a little bit of spinal fluid and you put some of these fibrils in and you just shake it for like a week, turns out that if you have Parkinson's disease, they're going to be able to see aggregates forming in this machine as you shake the spinal fluid for 2 weeks. And also you can do this from samples of skin or a submandibular gland, where there's also these abnormal synuclein fragments. And so we've studied it. A number of other groups have studied it. We've looked at all different techniques, and it looks incredibly accurate. So I think that within a short time, it's going to have to be commercialized. Now it's not commercialized, but this looks like it's the real thing, that we're going to be able to detect who has Parkinson's by measuring this abnormality of aggregation of synuclein in spinal fluid or other tissues.
Fantastic. That's a proof of how using the NINDS's resources can really make a massive difference. We have a couple of questions to change to the topic a little bit about COVID. So our first question was from Deborah about this. Is there NIH funding about research on COVID and neurologic diseases, specifically long haul symptoms of COVID and neurologic diseases, and then anything specific about COVID and Parkinson's disease that NINDS is funding?
Okay, well, if I go to the last question first. So in the beginning, well, even now, there's so many questions about COVID we don't know. But in the beginning, there were reports of people developing a Parkinson's syndrome with COVID. And that led to concern at, what happened in the influenza epidemic of 1918, where people with influenza strain got what's called encephalitis lethargica and then developed Parkinson's. People worry, is that going to happen? Well, the good news is no, it's not happening. There were a couple of cases where we think the inflammatory response to the virus actually caused damage inside the brain, caused symptoms of Parkinsonism, but not really, it's what's called a secondary Parkinson's syndrome due to injury to the brain. And in COVID and in other inflammatory diseases, this rare called acute necrotizing encephalitis can be seen in the spinal cord and the brain. And there have been definitely a handful to a dozen cases in COVID, but it looks like it's going to be extremely rare. So not something that's at pandemic size a problem like it was after the 1918 flu epidemic. And then in terms of the post-acute effects of COVID, what we do need to do is because it's affected so many millions of people, is set up to study whether could it have increase someone's risk of developing Parkinson's 10 years from now? Or does it decrease your risk of developing Parkinson's 10 years from now? Now, the COVID was a major stress on the body, and the brain can be involved. So those kind of questions we have to get at by doing large epidemiology studies, hopefully through electronic health records where we can do that now. And that's one of the components we call the post-acute sequelae of COVID, long-term effects. The bigger component that's most in the press is the symptoms that people who have COVID, suffered with COVID, are still suffering now months afterwards. So NIH is about to launch, we're actually talking about it this afternoon, a large project which was, again, appropriated funding from Congress, $1.15 billion to study this syndrome. We have plans to bring in cohorts of patients who are still having trouble to be studied very intensely, upwards of 10,000 patients with these symptoms to try and get at the bottom of it, along with these electronic health record studies to follow people over time, and then also autopsy studies to look inside if someone dies for whatever reason, to see what the effects of the virus were, whether there's still any virus present or viral components present that are triggering an immune response. So this is a big project, and we have a really ambitious way going about it, ready to launch.
That's excellent news. Greg asks a follow-up question, which is warp speed, which referred to how quickly the COVID vaccines were rolled out or created and rolled out. Could any of those practices apply to Parkinson's disease research in general, or particularly understanding COVID in Parkinson's disease?
Well, that's a good question. So the process that took place at NIH is what's called ACTIVE, and that was an industry, academia, FDA, all coming together and working on different components. So vaccines was one of them. I worked mostly on developing therapies for people who were sick with COVID, and it was pretty impressive how everybody got together and how fast things happened. So there were like ability to screen thousands of compounds to see which ones were effective against the virus. And then clinical trials across the country set up to test different agents in people who were sick at various levels with COVID. And this went at very, very quickly. If you think about, what are the effects, what are the lessons learned, I think it's one lesson learned is that, getting the industry people together with the academia people is really quite, really changes the game. We do that in the AMP-PD for Parkinson's now, and that's also been a game changer for us. But there's so many other areas besides the biomarker side of things that we could be working with the companies to attack this problem of understanding how to slow down the disease of Parkinson's disease. And then there's also a lot of companies that got started to adjust to COVID. So developing, very quickly therapies that affect certain genes. Those could actually help us in Parkinson's disease going forward.
Fantastic. So there's one additional potential silver lining of the COVID epidemic is potentially figuring out how to make medical breakthroughs happen a little quicker. So
let me just say, there's one other thing that I don't know if any entrepreneurs out there, but this assay I told you about, which takes weeks, you have to shake the spinal fluid for weeks to see if there's an abnormal synuclein. Well, I'd love to see somebody be able to develop a technology where you can detect that in a day, with some other type of assay, because there's something abnormal in that fluid. Why does it take a week? Plus, it's not quantitative, so you don't get you have a little bit or you have a lot. It's either a yes or no answer in that test. So the technologies that people are working on to detect COVID, it would be great if they could, say, well, here's a problem, figure out a way to detect abnormal aggregates of synuclein in the spinal, and that actually give you quantitation as opposed to a yes/no answer.
So lots more to be done, but the start is promising. So that's really great. We have time for one last topic to discuss, and many people in the chat are very concerned, the Parkinson's disease community in general, concerned about the environment, environmental triggers of Parkinson's disease. And Roger asked a question: when will the EPA finally ban paraquat and TCE, which have proven connections with Parkinson's disease? And does the NIH have any relationship with the EPA on influencing those type of decisions?
The NIH doesn't influence any other federal agency. I think if I tried to do that, I think I'd lose my job. We do is our job is to do the research that creates the evidence that then informs the EPA on what to do, as well as physicians. So we don't make guidelines and tell physicians how to treat Parkinson's. We present them with the data and they develop the guidelines. So it's not that much different. But I think the main point that you bring up is how to get a handle on what are the environmental influences that are triggering Parkinson's disease. And I kind of hinted at that when I was talking earlier, that we studied the genes that give rise to Parkinson's, but we're pretty much hitting the ceiling now. And we kind of know that the percentage of Parkinson's that can be explained by genetics, the most is going to be somewhere around 20%. And that leads 80% that must be environmental factors. And that's really the big challenge. So at NINDS, we're thinking of actually starting a new office, which would basically just concentrate on that problem, not just for Parkinson's, but actually Parkinson's is actually the one that's front and center. But all our diseases have that. We're running into that problem. And so we want to work with the National Institute for Environmental Health Services and try and bring the people who do epidemiology, say, so you're going to talk to Caroline Tanner next, I think, in the session. She's like world's expert. So that's a good question for Caroline. But what we want to do is to take the epidemiology data and try and understand at the neuroscience level, so we can either develop some kind of science about how in the environment leads to the synucleinopathy that causes Parkinson's, and then we can either intervene, with potentially a protective drug, or we could potentially say, we have to get this out of the environment because it's too toxic. And I know that Ray Dorsey has been really kind of pounding the pavement about the environmental aspects of changes in industrial society that led to, he thinks, Parkinson's disease. So we have a lot of culprits out there. It's hard to put one of them on the stand and convict them to a certain extent in terms of when you think about the entire Parkinson's population. But that's what we have to do. We have to try and identify these culprits and the real drivers. We have to convict them and get rid of them from our environment.
So that's excellent news, and I think it's going to be very heartwarming to people listening that you, as the director of the NINDS, know about Ray Dorsey and know about these various movements that are coming from the Parkinson's disease community, and you're responding to them, which I think is really absolutely wonderful. And this leads to our very last question, which is we have a large community, active community of people with PD who really want to make a difference and get involved. What are the things that we as a community can do to make our voices heard so that the national government knows about the importance of research in Parkinson's disease, knows about the importance of these environmental factors, for example? What are things that the community should be doing?
Well, I get this question quite frequently from different groups, and I may be off base, but what from looking from my vantage point, the fields move forward because really smart, dedicated, passionate people enter the field. And NINDS is not a great encourager of people. We are a federal agency. We're really boring. We review the grants. We give the money out, but we don't influence somebody to spend 180 hours a week studying Parkinson's disease. But you can do that. I think that's the power. I think of the patients and the advocacy community is to influence young people to study Parkinson's disease. I think we know, people who have just been giants in Parkinson's disease. And if you talk to them about why they got into it, it's usually because they met somebody with Parkinson's or they ran into the disease organization. And you can't underestimate the value of your organizations, patient organizations, for motivating the scientists. And in actual fact, they get a lot more fulfillment out of their career if they're aligned with the patient organization and if they're just doing this, in their lab all by themselves, not integrated into the greater community. So the great thing about the Parkinson's community is it's so close knit, but that's because of the patients and the patient advocacy groups. So anything you do to step that up, try and go to people who are younger, because the most impressionable ones are the youngest ones. They're thinking about, what am I going to do with my career? I have all the options. What should I do? And I think that's where the power of patients and advocacy groups is really underestimated with their value to the research.
That's a great point. And it really is something that APDA takes to heart, because that really is our research motivation. We really try to fund those first level grants that don't have enough results to actually even apply to the NIH yet. They get a little money from us. They can do their research and then get the results that then they can go to the NINDS with. So we really take that to heart here.
Yes, you do. Yeah, we really appreciate it. It really pays off.
Thank you so much. I just want to thank you, Dr. Koroshetz, so much for such a wonderful presentation. We're out of time. We can't ask any more questions. So thank you so much to everyone who participated, for all your wonderful questions, for all your thoughts about what's important to our Parkinson's community. And if you know somebody who missed today's program, if you joined late, if you'd like to watch this again, if you want to hear all of Dr. Koroshetz's amazing insights again, please feel free to go back to our YouTube channel. Later today, you will have access to the entire broadcast. And don't forget to subscribe to APDA's YouTube channel to watch new videos and live broadcasts. So for additional information and resources, please visit our website at apdaparkinson.org. And you can check out everything that we have to offer there. And I want to specifically mention that the next episode of Dr. Gilbert Hosts will be held on Wednesday, June 16th at noon Eastern Standard Time. I will be hosting Dr. Carolyn Tanner, which Dr. Koroshetz just mentioned. She's an expert on the environment in PD. And so we will be hearing from her and having a Q&A about the environment with her. Now, there's a lot of concern and connection about how exposures to our environment may increase the risk of PD. That came up on today's conversation, and we're very excited to bring you this conversation next month. And you can register for this session as well as our other virtual programs using our virtual calendar. If you go to apdaparkinson.org/events, scroll to June 16th, and you can register for that event. So we hope you enjoyed today's program. And if you did, we hope you will consider supporting APDA with a donation. Because with your help, APDA can help conduct more research, as we discussed, and can deliver more programs like this. And once again, I want to thank Dr. Koroshetz. I want to thank all of you for joining us today, and we hope to see you soon on another APDA program. Have a wonderful afternoon.